Okay, welcome to this podcast for cell biology. In this podcast, we're going to talk about mitosis. Remember, on that last podcast, we had taken our one cell here that started off with our two chromosomes, homologous chromosomes. It copied them during the S phase. It then went to the next stage of G2, where the cells got bigger. So that's G2, remember, and this was G1. And now what we need to do is take these cells and somehow make two daughter cells. Each that will look identical to this starting cell. And so this process here, as I said on the last podcast, is called mitosis. And so that's what we're going to spend the next several minutes talking about, is mitosis. Okay, let's get started. I want to first begin by listing the 5 stages of mitosis. The first stage is prophase. The second stage is prometaphase. The third stage is metaphase. The fourth stage is anaphase. And telophase is the last phase. And I want to go through each stage of these in a little greater detail, drawing what the chromosomes look like. But before we get started, I want to remind you what the chromosomes look like during interphase, during G2 in particular, before they hit mitosis. We didn't draw it on the last slide, but remember, this is all occurring inside of the nucleus. And I drew the chromosomes on the last slide in a very thick representation. But in reality, the chromosomes during interphase are going to be very thin, and they're going to be difficult to see under the microscope. So one of the first things that happens when the cells enter mitosis is that these chromosomes are going to condense. And so let's draw this first phase of mitosis, prophase. We still have our nucleus here, and as I alluded to, the chromosomes are going to condense. They're going to become much thicker, as I'm trying to show here. So let's write that down. During prophase, chromosomes condense. During G2, we should write here, chromosomes are not condensed. Another important thing happens during prophase, and that is the structures called the centrosomes begin to form. And what the centrosomes are is they are the microtubule organizing center. And so I'm going to draw them like this here. We're in the cytosol, but these are the centrosomes. And as they form, they will begin to migrate towards opposite sides of the cell. Okay, now we're ready to move to prometaphase. Okay, I redrew prophase here just so we could follow it as we look at prometaphase. In prometaphase, some of the main things that happen is that this nuclear envelope begins to break down. So as this nuclear envelope breaks down, these chromosomes are now in the cytosol. These centrosomes now will begin to send spindles out to attach to the chromosomes. And they attach near the centromeres at a place that we're going to call the kinetochore. The kinetochore is where the spindles attach. So they contain centromere plus proteins that are needed to attach these spindles. And remember, I called these purple lines here, all of the purple lines here, I called spindles. And the spindles are nothing more than microtubules. They attach to the chromosomes. And as you'll see in metaphase, they're going to move the chromosomes to specific places in the cell. So I'm going to erase these words here so we can talk about metaphase right next to it. Though I should have written down here what are the hallmarks of prometaphase. I said it, but let's write it down. Nuclear membrane breaks down. Spindles form and attach to kinetochore. So let's talk now about metaphase. During metaphase, what happens is the chromosomes line up in the middle of the cell. And they do so in such a way where one sister chromatid is on either side of the cell. Now, one thing I haven't been showing as I've been drawing this is a cohesion molecule. So I do want to come back to that because it's important at this stage to talk about it. Even though I've been showing these chromatid separated here, remember, they're not really separate. They're still being held together, same here, by this protein called cohesion that we talked about earlier that forms during S phase. It's over here too. It's important that it's still forming here and still holding them together, because if it wasn't holding these sister chromatids together, you could easily end up with both of these sister chromatids on the same side of the middle of the cell. So how did these chromosomes end up right here in the middle? Well, it didn't just happen by accident. It happened because these centrosomes had originally extended these microtubules across the cell to attach to each of the sister chromatids. And what happens during metaphase is after they've attached, they begin to retract some in such a way that they position them right in the middle of the cell. So that's the whole job here of these spindles. They reach out, they grab the chromosomes, and then they pull them to the middle of the cell in such a manner that one sister chromatid is on the left half and the other is on the right half. Now remember, it's much more complicated than what I've drawn here, because what I've shown here is only one set of homologous chromosomes. Remember, in a human cell, there would be 23 pairs of chromosomes lined up along here, for a total of 46 chromosomes. But this is just a more simple way to look at this. So at the end of metaphase, these chromosomes are now nicely lined up in the middle. I'm going to erase this half of the board now. Not so I don't have to redraw this. I'm just going to extend the process of mitosis and just draw it going this way. So now we enter anaphase. In anaphase, we're going to pull these sister chromatids apart from each other. Remember, during metaphase, what's happening is that these spindles here are pulling on these sister chromatids, but they can't separate them because cohesion is holding them together. What happens during anaphase is the cohesion breaks down. By it breaking down, now these spindles can successfully pull each component of the chromosome to the other cell. Cohesion's gone, but these centrosomes are still here. With the spindles attached to them, they are now pulling these sister chromatids apart from each other. And we'll write down here what's happening up here. These chromatids, the sister chromatids, pull apart from each other. Now what happens is we're going to enter into telophase. Because what's now happening during telophase is that the chromosomes are on opposite sides now. And the nuclear envelope reforms. So let's draw that. So we've pulled our chromosomes successfully to the other side of the cell. They are separated. And now the nuclear envelope will reform around them. At this point here, the centrosomes will break down and the spindles will have released their hold of the chromosomes because they're no longer needed. Towards the end of telophase, the chromosomes will decondense, so they're going to be less visible. So we're going to redraw them to illustrate that. So as best as I can, I'm trying to show them in a less condensed state. Now remember, in humans, this is more complicated. We would see 46 chromosomes in each of these nuclei that just formed. Or you could say 23 pairs of chromosomes. All right, now what happens? Well, what happens next is that this cell at the end of telophase will begin to pull these membranes, the plasma membranes, together, and eventually it will pinch them off like so and form these two new cells with their decondensed chromosomes. And this is called cytokinesis. We call these two cells daughter cells. And if you remember from several whiteboards ago, when we began talking about the cell cycle, we began with a cell that looked similar to this. Going through G1, S, G2, and now meiosis, we've successfully copied these and segregated them, the copies, into two new cells, such that they are identical to each other. Okay, that is all I want to say about mitosis. But now I want to move on and just spend a little bit of time talking about how bacterial cells divide and produce daughter cells. So this is called, as I mentioned before, binary fission. It's more simple than eukaryotic mitosis, mainly because bacteria have fewer chromosomes, usually just one, and they lack a nucleus. And related to this first one here, they have less DNA. So let's start with this cell up here, this bacterial cell, and we'll draw a chromosome in it like so. It will copy the DNA in a manner similar to how eukaryotic DNA is copied, and you end up with two chromosomes. And I've drawn these as circle because most bacterial chromosomes are circular. Though bacteria don't have microtubules, so they're not going to have those centrosomes, they do form these filaments that will reach out and attach to the copied chromosomes, and they will pull them in opposite directions. until they reach the opposite sides of the cells. And then what happens is they begin to form this ring of proteins that bring these two plasma membranes close to each other. So you end up with something that looks like this, with this ring forming around, pulling these two membranes near each other. At some point, these two plasma membranes fuse with each other, and you end up with two separate bacterial cells, each containing a copy of the DNA. You can see it's similar to what happens in eukaryotes. You copy the DNA, you form a fiber that's not a microtubule, but it functions similarly, that now will pull those chromosomes to opposite ends of the cell. And then, by a mechanism similar to cytokinesis, but not the same, it will pinch these two plasma membranes together until you have these two new cells. Okay, well that's all I have to talk about today. We've talked about the cell cycle and mitosis, and then ended here with how bacterial cells divide and copy their DNA. But if you have any questions until then, please make sure you let me know. If not, I'll see you in class. Bye.
Not Sheldon. Over the years, we formulated many theories about how he might reproduce. I'm an advocate of mitosis.
I'm sorry?
I believe one day Sheldon will eat an enormous amount of Thai food and split into two Sheldons.
On the other hand, I think Sheldon might be the larval form of his species, and someday he'll spin a cocoon and emerge 2 months later with moth wings and an exoskeleton.
Okay, well, thanks for the nightmares.
More pad Thai, please.
Sheldon, you've already had 4 servings.
You might want to slow down a bit, buddy. Just one more bite.
Sheldon? Sheldon, are you okay?
I'm just so full.
That's it. No more Thai food.